Hi, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the beautiful inland Pacific Northwest. Today is the 4th of June 2020, and as promised, I want to get back into intermediary metabolism. Specifically, I want to talk about bioenergetics. Now, rather than talk just about ATP and how energy is necessary to mount, for example, an immune defense, or to respond to changes in diet, or to respond to changes in deficiencies. I'm going to give you a really sharp focus on one particular, what I call rheostat, that specifically works on regulating whether or not lipid metabolism is turned on for biosynthesis or for oxidation of fatty acids, and therefore for the production of ATP. Because, of course, when you oxidize a fatty acid, you're going to reduce both nicotinamide adenine dinucleotide and flavin adenine dinucleotide to the reduced forms, that's NADH and FADH2. Those are going to be run through the electron transport chain so that they can become reoxidized, and then in the final process of that 5-complex system in the inner mitochondrial membrane, generate ATP from the oxidation of fatty acids because of the production of NADH and FADH2 each time one of the oxidations occurs. So this is far more bioenergetically encouraged in the cell, and also enriching for the system, because you get a great deal more ATP per carbon-carbon bond by oxidizing a highly reduced form of hydrocarbon like a fatty acid, which is actually an aliphatic monocarboxylic acid, as opposed to something like a carbohydrate like glucose. So I think we've all heard that in the past, and so I'm not going to belabor it again. But let's jump into this, I think, really fascinating story. So again, I'm Dr. Dan Guerra. You can see me there. And as soon as I get this thing set up, we will start at the beginning, which is usually a good idea. Okay, so let me hide this little unprincipled bar down here. There we go. So, as the title suggests, bioenergetics and intermediary metabolism, biochemical means to a physiological ends. So the foundation of everything that happens physiologically has to happen biochemically first. And so, in fact, the biochemistry weaves in and out of the physiology all the time. Otherwise, there's no physiological presentation at all. So again, I think that's not just because I'm a biochemist that I think that way. I think really the foundation of any kind of cellular system, before you have cells, you have to have chemistry and physics involved. And because it's a biological system, it's going to be biochemistry and biophysics. Now, biophysics, of course, is playing a role in like cell structure, function, morphology, growth and development. But that's not what I study. I study the biochemistry. And so you're going to get that point of view and that link directly more into the requirements for normal physiological growth, development, and health than you are from a biophysical principles discussion. But both of them, I think, would be reasonable to discuss at some time. So, again, I'm Dan Guerra. So here's the little conduit I want you to look at. It is whether or not you incorporate carbon dioxide via acetyl-CoA carboxylase, here, this enzyme, taking carbon dioxide from the air and acetyl-CoA from metabolism, and synthesizing Malonyl-CoA, a C3 intermediate, for what? For the synthesis of fatty acids and all lipids. Malonyl-CoA is also involved in many other important pathways, which we can get into as time goes on. Right now, I'm going to look at particularly the production of fatty acids and or their oxidation. Now, interestingly, Malonyl-CoA, where it can be metabolized out into making long-chain polyunsaturated or monounsaturated or saturated fatty acids for that matter, as CoA esters or as intermediates of the acyl carrier protein fatty acid synthase complex, still a thioester, of course, still a phosphopentathiene prosthetic group like coenzyme A, but not a freely moving molecule like a CoA thioester, but one bound to a backbone of a protein. Rather than use mal-CoA for that, mal-CoA can be quickly decarboxylated. So that looks like almost a futile cycle, but it certainly isn't, because it's not happening at the same time, right? You don't go through all the effort with the acetyl-CoA carboxylase enzyme, which is a biotin-containing enzyme. That whole process is very well regulated, orchestrated in the cell to synthesize malonyl-CoA. You don't simply make malonyl-CoA just to break it down, obviously. So there's got to be a reason why there's an active malonyl-CoA decarboxylase. And I'm going to explain that to you today. Now, before we get into that, here's a paper, Journal of Visual Experimentation, 2009, October. You can see the citation here. And I want you to listen to follow me here because it's going to lead into this really cool discussion today. So there are two different methods for creating a myocardial ischemia, and they're both used in the rodent model. Ischemia is going to lead to an infarction, right? Be blocking blood flow, and therefore the ischemia part of it is the lack of oxygen, right? So there's the cryoinfarction that's used in rodents. It's fast, but it's an inaccurate technique, and it uses a cryopen as applied on the surface of the heart, leading to a variable, so it's not absolutely precise because you're using a pen, cryoscar. Now that can lead to ischemia, however, with considerable collateral myocardial injury, as you might guess. And that could confuse the pathophysiological sequelae with discrete and monospecific myocardial infarction being the goal. So there's a far better surgical method. And this surgical method is actually a permanent ligation of the left anterior descending artery, or LAD, where LAD is ligated with one single stitch. This is the rodent model, of course, forming an ischemia that can be seen almost immediately by monitoring, right? By closing the LAD, no blood flow is permitted, while surrounding myocardial tissue is not affected. That's the reason why it's a better technique. Therefore, it's preferred technique as the surgical procedure mimics pathobiological and pathophysiological aspects occurring in an infarction-related myocardial ischemia. Now, that's so that you understand the next slide. So, pharmacological inhibiting of the malonyl-CoA decarboxylase in post-MI rats using that surgical procedure decreases the severity of pre-existing heart failure post-MI. Okay, so that's what this experiment's based on. All right, so let's take a look at this. So MCD inhibition is able to immediately improve cardiac function in the failing heart. And that suggests that there's an optimizing cardiac energy metabolism since this decarboxylase is involved in energy metabolism. And maybe it acutely improves heart function, at least in this ischemic response. So we'll see. Oh, by the way, this paper is from the Journal of American College of Cardiac Basic Transactions in Science, published just last year, 2019. So that's where this paper is coming from. The last one was 2009. This one was 10 years later, so just about, not even a year ago, 6 months, 7 months ago. So, both immediate and chronic inhibition of MCD improves cardiac contractility in the failing heart. That's associated with an increase in mal-CoA content in the post-MI heart, resulting in a decreased cardiac fatty acid oxidation rate. Okay? Now, I'll explain all that in a moment. MCD inhibition improves cardiac efficiency. Here's the physiology, or the correction of the pathophysiology. It improves cardiac efficiency that is associated with a reduced proton production through increasing lactate dehydrogenase isoform 1 activity after its expression from transcription, while decreasing an isoform of LDH called LDH5. Okay, now I want you to keep that in mind. Now, I put these little asterisks up to remind me. Lactate dehydrogenase 5 is an isozyme of that enzyme composed of 4M type polypeptide chains. It catalyzes the conversion, of course, of pyruvate to lactate with an unparalleled efficiency. So it's anaerobic oxidation, right? So dehydrogenase, that's what those do. But this function of LDH gradually fades away as number of the H monomeric subunits over the M chains increases. Thus, LDH1, another component isoenzyme made up of 4 H polypeptide chains, okay, favors an aerobic oxidation of pyruvate via what? Pyruvate dehydrogenase. Of course, that's the enzyme that leads into the TCA cycle. So take a look at this. So you've got this LAD ligation. We just saw what that was, remember? I just explained that to you. Here's the rat heart. You induce a myocardial infarction, which is, of course, from an ischemic response. And then 3 weeks after that, now you're looking at the appropriate effects on that on heart and heart's ability to continue to function correctly. So you're looking at whether or not heart failure is going to be exacerbated by this response. You add a malonyl-CoA decarboxylase inhibitor at 4 weeks, or you have the vehicle. When you add the decarboxylase inhibitor, you tank fatty acid oxidation, you enhance cardiac efficiency, stroke efficiency, and you increase cardiac function, okay? At the same time, if you're using just the vehicle, the opposite happens. You get increased fatty acid oxidation after ischemic event. You get a decrease in efficiency, and you get, of course, a decrease in function. So what they think is happening is shown here. So you've got malonyl-CoA, okay, forming acetyl-CoA normally by the decarboxylase, but we're blocking it. So that means mal-CoA is going to build up. When malonyl-CoA builds up, what's function is, in this kind of system like the cardiac muscle, is it blocks CPT1. That's carnitine palmitoyl transferase 1. So it will block the uptake of fatty acids into the mitochondria, and therefore it will inhibit fatty acid oxidation. What that then will induce for the alternate carbon source is cause glycolysis to carry out anaerobic glucose oxidation. It's going to produce protons in that pathway, and that's going to enable more ATP synthesis. That's what this is supposed to be suggesting here. And therefore, because of the slight acidification, it's also going to enhance contractility of the muscle. So you get a tanking of fatty acid oxidation, where you get an increase in glucose oxidation, okay? And then ultimately, acetyl-CoA is formed, of course, if it goes through the pyruvate dehydrogenase, but that depends on which way it goes. So if you were to take pyruvate and convert it via pyruvate dehydrogenase, right, which of course makes acetyl-CoA, and that's acetyl-CoA, which would be an actual oxidation now, because you're going to reoxidize the NADH made from the running of the acetyl-CoA through the TCA cycle, then that's going to be similar to beta oxidation. But if you just use glycolysis, or the anaerobic mode, you don't run the TCA cycle. And apparently, after an ischemic attack, where oxygen has been deprived, you get better function and better efficiency for cardiac muscle repair and functionality, and finally then a lack of heart failure subsequent to the myocardial infarction if you use glucose rather than fatty acid. Okay, that's what this whole paper says. Now, a paper published way back in Tumor Biology 2003 says the following. This is added to this, okay? LDH1, so if you want to know the difference in the two isoforms of the lactate dehydrogenase, and of course you do, because this is authentic biochemistry, not kiddie biochemistry, so it's not textbook. LDH1 is found in all cells, while LDH5 is preferentially found in tumor cells. That's interesting. While normal tissues were practically devoid of LDH5. So interestingly, LDH5 positive stromal cells are associated with hypoxia-inducible factor 1 alpha overexpression. Okay, now what all this means is it's concluded that normal tissues utilize aerobic oxidation, okay, normal oxidation, as a means of energy production, while tumor cells are turned into anaerobic glycolytic systems. That's a phenomenon which is rarely followed by the mesenchymal cells of a tumor-supporting stroma, which normally likes to use fatty acids and fatty acid oxidation. Okay? So this is a really important thing. LDH5 is preferentially found in tumor cells. We'd already told you that LDH5 catalyzes the conversion of pyruvate to lactate with unparalleled efficiency, right? So that means you're going to get anaerobic oxidation, right? So LDH5 is preferred in the tumor cells. So that means you're going to get a lot of what? Glycolysis. That's known as anaerobic or rampant anaerobic glycolysis in the tumor cell. That's totally different when you think we switch from tumor pathology, pathophysiology, to myocardial infarction physiological requirements, right? Here, glycolysis is preferred. So depending on the two diseases, cardiac disease versus cancer, the biofuel switching is really important. And you can tweak one system or another, for example, LDH1 or LDH5, via the MCD pathway, and thus enhance, if you want to enhance the glycolytic efficiency by blocking fatty acid oxidation in the ischemic heart right after an infarction, maybe inhibiting the malonyl-CoA decarboxylase would work, because then you get a buildup of mal-CoA, and then you don't get beta oxidation because you block, because of the increase in mal-CoA, the uptake of fatty acid in the mitochondria via CPT1. Get that? Okay. Really important pathophysiological consideration there. Sorry. Now, I want you to keep in mind this. Back to hormones. Glucagon and epinephrine work through their own receptors, right? A beta-adrenergic receptor for the epinephrine, and glucagon its own receptor. Both of them would turn on adenylate cyclase, which increases cyclic AMP, which would then activate, because it's a cyclic AMP-dependent protein kinase, A. That would then phosphorylate a series of enzymes, and it would do the following. It's intermediary metabolism. So it would enhance glycogen breakdown. It would enhance glucose synthesis. Because glucagon, of course, is the hormone that is sensing a limitation of glucose in the blood. But it would block glycolysis, because that would be a futile cycle of gluconeogenesis and glycolysis at the same time. In the same cell, you get the point. It also is going to tank lipogenesis, okay? So that phosphorylation cascade from those two hormones is going to control at that level. Now, what I just gave you was what happens downstream from hormonal control, right? At the enzymological level, at the cellular level, what's going on with bioenergetics. And we're going to continue on this discussion. So I want you to keep this in mind, okay? Really an important thing. Now, you will recall also that there's another kinase called the AMP kinase. That shuts down ATP requiring processes, and it stimulates ATP producing processes. Now watch this. So for example, after hypoxia, such as an ischemic attack, or with a lot of excessive work, like cutting wood or something, or splitting wood, you're going to get an increase in AMP because you're breaking down all the ATP, right? ATP to ADP to AMP. You're removing phosphoryls during work or because of hypoxia. So that turns on the AMP kinase. The AMP kinase is turned on by AMP. Now what that will do is also phosphorylate enzymes. One of the processes that will be kicked on because of this particular phosphorylation is glycolysis. So you're going to run some glycolysis, you're going to run all the carbohydrate you have on hand, through glycolysis to make pyruvate. You're also going to enhance fatty acid oxidation switch. You're looking for what? You're looking for energy. It's the ADP producing pathway. Now, at the same time, other enzymes will block gluconeogenesis. You don't need more glucose. You need more ATP. Protein synthesis, you don't need more proteins. You need ATP. Lipogenesis, very energy requiring pathway, an anabolic pathway. You don't need more lipids. Same thing with cholesterol, which is another lipid. Okay? So that's how these systems work. They're working in tandem, and they're set to be poised according to nutritional needs, the lack of oxygen or the abundance of it, stress, or whether or not there's a pathology involved, such as cardiac ischemic attack or such as the growth of a tumor. This is what I'm trying to put together for you. The fact that these systems are poised, and they're not simply poised via all this or all that. They are in a non-limiting Liebig's law sort of requirement. So Liebig's law, the minimum, which I learned when I was an undergraduate at the University of Illinois a few years back, basically said that a system can only function at whatever subsystem or event becomes limiting. So you think about the staves of an old barrel, and you think about the staves of a barrel at the top and how they're pushed down and the barrel is made. Right? If one of the staves is pushed down too far, like at the top, that one that's pushed down will leak off from all the other ones that are still in place, whatever you filled into that barrel, right? Like, say, putting in water or something else. It'll leak out. So that minimum, wherever that minimum level is, and there is just a level of where the stave is in a barrel by a barrel maker, right? That's what's going to limit how much volume you can get into that barrel. That's what that means. Law of the minimum. All right. So, yeah. So let's look at this here, for example. So you got glucose. And when you're in a well-fed state, what goes on with glucose metabolism? You run glycolysis. That's what this is here. Fructose 6-phosphate, fructose 1,6-bisphosphate, to PEP, to pyruvate, and then pyruvate going into the mitochondria, generating acetyl-CoA, that's the PDH pathway, or the pyruvate carboxylase pathway making OAA. They combine to make citrate. Okay, this is the well-fed state. Now, while that's happening, what else is going on is that there's an alternative pathway for glucose oxidation, and that's the oxidative pentose shunt. Here you make NADPH from turning glucose 6-phosphate into 6-phosphanogluconate, and then ultimately, after the loss of CO2, another mole of NADPH is made, this is 6-phosphogluconate dehydrogenase, making another mole of NADPH. So you're making a lot of NADPH. That reducing power is going to be used to drive what? It's going to be used to drive fatty acid synthesis. That's correct. And plus, it's going to be making a lot of intermediates important for driving anabolism. Ribulose 5-phosphate can be converted to ribose 5-phosphate, which of course is necessary for nucleotide biosynthesis. So look what happens. You get all this NADPH. So the pyruvate that's run from glycolysis, right, is going to be then converted to acetyl-CoA and OAA, citrate. Citrate will leave the mitochondria. This is not a business of making a lot of ATP. It's a business of making what? Lipid. Right? Citrate leaves make acetyl-CoA, also OAA, because that's what it was synthesized from. The malic enzyme, after malate dehydrogenase reaction, the malic enzyme will also make some NADPH and make resynthesized pyruvate, driving the carbon back into the mitochondrion because there is a transporter system, a monocarboxylic transport system for citrate versus pyruvate. And because of that, you're able to use the carbon to drive more lipid synthesis, see? So that's the fate of the OAA to malate to pyruvate and back down into this system. But the acetyl-CoA, you can form cholesterol via the prenolipid pathway, going through acetoacetyl-CoA, and ultimately mevalonic acid pathway to cholesterol 29 steps later. Or you can go through and make malonyl-CoA, excuse me, via the acetyl-CoA carboxylase. No, I'm not going to cut that out. I sneezed, sorry. So malonyl-CoA then is used as the 3-carbon precursor to do the condensing enzyme at the beginning of fatty acid synthesis on the way to making C2 to C16 fatty acids in the de novo FAS pathway. All of that requires a lot of NADPH. Cholesterologenesis, you need cholesterol to make new membranes, for example, for cell division, and fatty acid synthesis, because that's all your phospholipids, and also that's going to help synthesize all the sphingolipids. So the membrane lipids are going to be made and from fatty acid, the acyl membrane lipids, as well as all the prenolipids. All right. So that's why I showed you that pathway. Okay. So that's explaining to you what the acetyl-CoA is being used for here, right? And how malonyl-CoA is important for making fatty acids and thus driving membrane synthesis and also potentially, excuse me, triglycerol biosynthesis. All right. Now, let's take a look at what insulin does. We've looked at this before. Insulin shuts off something called the forkhead transcription factor, which would normally activate gluconeogenic and fatty acid oxidation genes. Okay, that's the IRE genes. That's the insulin pathway, right? So if it blocks forkhead transcription, if forkhead transcription normally turns that on, right? What's going to happen with insulin binding to its receptor, right? It's going to shut down gluconeogenesis. It's going to shut down fatty acid oxidation. That's what's going to result. At the same time, insulin is going to turn on the SREBP pathway. That's the steroid response element binding protein pathway, 1C. And that's going to bind to the steroid response element, and that's going to generate at the transcriptional levels, this transcriptional control now by insulin, all the genes that are going to enhance lipogenesis, the ones you just saw, fatty acid synthesis, cholesterologenesis. Those enzymes are also all going to be needed to be transcribed to kick on for anabolic pathway. This is the other thing that insulin does. This is all intermediary metabolism at the transcriptional level, initially sensu stricto. Now, glucose uptake is also going to occur here. And when glucose is taken up, there's also a binding protein, a carbohydrate response element binding protein, so the CH stands for there. And that's going to bind to its carbohydrate response element right here, which is a promoter region in lipogenic genes. And that's also going to enhance lipogenesis. So glucose enhances just directly through its receptor system is going to enhance at the transcriptional level lipogenesis, because that's just major storage form of carbon in a human, as well as insulin. They're both going to do the same thing, transcriptional level. And insulin is going to block this whole fatty acid oxidation, and we don't need any more glucose, so it's going to block synthesis of glucose. You get it. Again, glucagon, what it's doing at the transcriptional level now, turns on adenylate cyclase, turns on pKa. pKa is going to work through the cyclic AMP response element binding protein, or CREB. CREB is going to activate Cre genes. That's the cyclic AMP response element. At the promoter region of genes, those are going to be gluconeogenic, and you're going to enhance gluconeogenesis. You're going to make more glucose with glucagon. Glucagon means it's telling the system, make more glucose. Glucose is deficient. Hypoglycemia, right? Turns on glucagon synthesis in the alpha cells of the pancreas. Same time, it's going to block the Cre genes we just looked at. So it's going to block whatever lipogenesis is occurring. So this is yet another hormonal rheostat. If glucagon is tuned up, you need more glucose. You don't need to make more lipid. It's a waste of energy. And so gluconeogenesis is turned on. Remember, fatty acid oxidation is also going to be turned on in the glucagon mode of action. And because of that, you're going to get sufficient amounts of NADH and FADH2 to drive ATP synthesis in the mitochondria, and you need ATP to synthesize glucose from non-carbohydrate precursors. So that's the way that works. Again, it's a fundamental intermediary metabolism bioenergetic pathway by looking at transcription linked to hormone. Right. Fatty acids, on the other hand, are going to turn on the peroxisome proliferator activator receptor, alpha, in this instance showing it. What's that going to do? It's going to turn on the PPR element for mitochondrial FOX genes. Those are the fatty acid oxidation genes. It's going to turn on peroxisome fatty acid oxidation genes. It's another really important pathway because that's going to do partial degradation of fatty acids. It's going to allow them to reside in the peroxisome, and because of that, it's going to allow for retailoring of the membrane, phospholipid component. And it's also going to turn on ketogenic enzymes at the transcriptional level. So you're going to turn on so PPRE has 3 different gene sets it works on: mitochondrial beta oxidation genes, peroxisomal beta oxidation genes for membrane retailoring, and for removal of peroxidized fatty acids that can happen in membranes when they long live, when they have a lot of double bonds. It's also going to turn on ketogenesis. Guess what you're going to do all with the carbon, right? Because you're going to make all that acetyl-CoA. You have to make acetoacetate, beta-hydroxybutyrate, and get it out to the cells. Okay. Now, let's go back here. Yeah. So now we're back to Malonyl-CoA decarboxylase. Here's the enzyme. There's the EC number. I talked a long time ago what EC numbers mean in enzymology. I'm not going to do it today, but those numbers designate the kind of protein it is as a catalyst. But when you get deficiency of that enzyme, so that's called MLY-CDD, and you can find that in the OMIM. Now, I'm going to explain all that in a moment, what that's involved in. That is a website that tells you about inborn errors of metabolism. Okay, so that when you get a deficiency like that, it's been mapped in humans, and it's a rare autosomal, which means it's not in one of the chromosomes. It's not an X or Y chromosome, so it's a somatic chromosome. It's recessive, of course, so somatic, autosomal, recessive inborn error of metabolism is a deficiency of MCD. The expression of decarboxylase is highest in cardiac muscle. Makes sense, right? Because of that rheostat control I showed you. Followed by skeletal muscle, same thing. And then, of course, in the brain, small intestine, liver, pancreas, and even kidney. Malonyl-CoA is the key intermediate for de novo fatty acid synthesis, as I just showed you. And its accumulation helps regulate beta oxidation via the inhibition of carnitine palmitoyltransferase 1, 51, which occurs as 3 distinct polypeptides, actually, CPT1A, B, and those are expressed predominantly in muscular cardiac tissue, and then CNS-associated carnitine palmitoyltransferase 1, C. Okay. Now, it's noteworthy that Malonyl-CoA differentially inhibits those 3 isoforms, as with CPT1B expressed predominantly in muscle tissues where the synthesis of fatty acids is negligible. So basically, you always got that isoform, right? It's 100-fold more sensitive to Mal-CoA concentration in the muscle, suggesting a predominantly regulatory rather than a synthetic role for the Malonyl-CoA in skeleton and heart muscle. That means you're not making fatty acids a lot in those 2 organ systems. And that's well documented. So in this instance, slight alterations in Malonyl-CoA will dramatically affect substrate consumption and energy supply in the heart and muscle, of course. A deficiency or inhibition of the decarboxylase could force accumulation of cytoplasmic Malonyl-CoA, thus blocking long-chain acylcarnitine acetyltransferases, resulting in an impaired fatty acid uptake and beta oxidation, which is following that, in both mitochondria and the peroxisome. Okay, so that's not a good thing to have a deficiency in, especially in some systems, some organ systems. Now, what OMIM is Online Mendelian Inheritance in Man. Okay, it's a website that's developed for people to look up inborn errors of metabolism, to look at what happens when you get deficiency in a gene in a human disease. So there's a particular entry for the decarboxylase. That's the one we're talking about right now. Results from either an homozygous or compound heterozygous mutation in the MLYCD, okay? And that's the name of the gene, right? The decarboxylase gene. And that's the number of it on that chromosome 16q23. This deficiency was first described in a paper published not that long ago, 1984, that concerned a 5-year-old boy who presented with short stature, abdominal pain, chronic constipation, episodic vomiting, very sick child, metabolic acidosis, elevated urine malonic, methylmalonic, and succinic acids. So it looks almost like an amino acid deficiency thing, but it's really a decarboxylase issue because some amino acids are decomposed to malonyl-CoA. Parents are first cousins, and a SIB had died at 3.5 months of age. So that tells you something. Child's mitochondrial malonyl-CoA decarboxylase was reduced while both parents had an intermediate level. Okay? So that's why it's homozygous recessive. All right. Again from here, subsequently analysis of 7 reported cases of MCD deficiency. Malonyl aciduria, methylmalonyl aciduria, because that's coming from the amino acid degradation pathway, and development delay seizure disorder, hypoglycemia, and cardiomyopathy. Finally, a report cited in the OMM, there's your citation, published not very long ago, in a cluster of patients resulting in 1 death of them from cardiomyopathy at 9 months of age. 3 of the 9 had seizures, which resolved over time in 2 of the individuals. 7 of the 9 development delays. 6 of the 9 had cardiomyopathy, most of which were detected via screening echocardiogram of 7 individuals who had brain MRIs. 4 had T2 hyperintensities in the white matter and basal ganglia. So what does that mean relative to this MRI? Okay, so now I'm going to explain that to you. All right, so this is coming from the Handbook of Clinical Neurology. So this is time to put on my glasses. This is showing you an axial MRI view. It looks at the brain from below, like from the chin, and a series of images starting at the chin and moving up to the top of the head. That's known as an axial image. A sagittal MRI looks at the brain from the side. It's a series of images starting from one ear and moving all the way to the other, like through the brain. That way, okay? Like through this part. The coronal MRI looks at the brain from behind, starting from the back of the head, and then moving all the way to the face, moving through. That's how that MRI works. This is just showing you those same images a little bit clearer. So what are we talking about here? This comes from the Handbook of Clinical Neurology. This is available online for free for all neurologists and for anybody who wants to look at it, actually. White matter hyperintensities, known as GWMHs, are initially clinically silent, but upon scanning, visible in deep or periventricular white matter on CT or MRI. They are particularly apparent on FLAIR MRI. Don't worry about that, I'll explain in a minute. Which is a T2 weighted sequence where the CSF signal is suppressed. Okay, so the cerebrospinal fluid signal is suppressed because you need that, otherwise it blocks the view. Owing to an increased water content and molecular degeneration in damaged white matter, the WMHs appear bright on FLAIR. Okay, so white matter hyperintensities appear white on FLAIR, making this a current standard imaging sequence of choice for determining those white matter hyperintensities. The underlying pathology is nonspecific, but includes demyelination, gliosis, and axonal atrophy. Three really unfortunate problems in the CNS. So WMHs are associated with stroke risk factors and are thought to be related to small vessel ischemic pathology, or more general vascular disease. WMHs are common in older adult brains and have been associated with advanced aging as well. So MRI provides fine neurological detail of the brain, as shown here. Spinal cord can also be analyzed, and indeed the vascular anatomy. And it makes one able to visualize anatomy in all 3 planes using MRI: the axial, the sagittal, and the coronal. Now I'll explain to you. Okay, now I want you to understand MRI is based, I want to explain a little about MRI. Right? So this equation is the one that's used for Fourier transform. So you can see this is a summation of 1 over n, with n going to 0, and with n minus 1 to n going to 0. And then this xn is going to be taken to this particular power here. Now, what does all this mean? So to find the energy, those are the purple terms in that equation. At a particular frequency, those are the green terms. Spin your signal around a circle at that frequency. This is what MRI is going to do. And average a bunch of points along that path. That's how the Fourier transform mathematics works behind the MRI. Now, follow me along here. MRI is based on the magnetization properties of atomic nuclei, where a uniform external magnetic field is deployed to align the protons of water, the most abundant fluid there, that are otherwise stochastically arranged. So this is going to basically polarize those protons, okay, via a magnet. After a uniform alignment, which is known as magnetization via the dipole moment of the water molecule, now it's been centered and fixed by the MRI signaling of the magnet. Once that's achieved, it is subsequently disrupted by the introduction of an external radio frequency, or an RF. As the nuclei return to their resting alignment, through physical chemical relaxation, RF energy is emitted and measured. And that's what the MRI is sending out to be run through a Fourier transform, which I just explained to you there really quickly. And then ultimately you get those images. You see. As the nuclei return to the resting alignment through physical chemical relaxation, RF energy is emitted and measured, as I said. Fourier transformation is used to convert the frequency information contained in the signal from that RF from each location in the imaged plane to corresponding intensity levels, okay, which are then displayed as shades of gray in a matrix arrangement of pixels. That's what we just saw in those images, those MRI images. By varying the sequence of the RF, again, it's the radio frequency, the frequency of those pulses, applied and then collected, right, because you're collecting the data, different types of images are created, okay? Repetition time, the TR, is the amount of time between successive pulse sequences applied to the same slice of tissue you're running through. Time to echo, or TE, is the time between the delivery of that initial radio frequency pulse and the receipt of the echo signal, which is subsequent. Okay. Tissue is analyzed via two different relaxation times, T1 and T2. See, I told you this is a T2. I got all this for you so you understand what T2 stands for. Those of you who are physicians and those of you who are just curious. T1 is a longitudinal relaxation time. It's the time constant which determines the rate at which the excited protons return to equilibrium. It is a measure of the time taken for spinning electrons to realign to that external magnetic field. Because remember, there's a dipole moment. They realign to that dipole, right? T2 is a transverse relaxation time. That's the time constant which determines the rate at which the excited protons reach equilibrium or out of phase with each other. Okay? It's more a dynamic system. T2 is more dynamic. So it's a measure of the time taken for spinning protons to lose phase coherence, lose that magnetized dipole structure among the nuclei which are spinning perpendicular to the main field. That's why T2 gives you more detail. Okay? Because it's measuring an event in real time. Okay? It's my way of describing it anyway. So biomedical features of patients, MCD, okay, going back to now the disease that the MRI is used for. Again, it's an autosomal recessive. It's not the gametes. Although it can be inherited, it shows up as autosomal recessive. It's not in X and Y chromosome, I should say. So you get a low-fat, high-carbohydrate diet obtains to near normalization when you give people that diet. This is one of the cures or remedies. You cure or normalize urinary organic acid excretion with no further hypoglycemic episodes. Hospital admission during times of infection as well as during febrile illness is usually important because of the problem with the immune system. Okay. One patient sample suggested a 4 base pair deletion of a 3' end of exon 2 in the gene, of the MLYCD gene, which is homozygous for MCD. In a larger study, there are 16 of 18 pathogenic alleles in 9 unrelated patients with MCD. Okay. Those deletions produce a protein. I'm just explaining to you now what they've been reported in the literature. They produce a protein that is non-functional. Hence, glucose becomes essential since fatty acid oxidation is almost completely blocked due to the blockage of CPT1 by the elevated malonyl levels. So that's where all of that comes from. Okay. So the diagnosis of malonic aciduria involves elevated levels of malonyl carnitine, also a C3DC in the blood. So you get a deficiency in the malonyl-CoA decarboxylase. You build up mal-CoA. Mal-CoA blocks the CPT1 that blocks uptake of acyl-CoAs to acyl-carnitine into the mitochondria. So the malonyl-CoA will degrade, though, okay? Or it can be used for fatty acid synthesis. When it degrades, it makes malonic acid, which further inhibits the TCA cycle, okay? So this is how it presents ultimately. The urine organic acid analysis shows the high levels of malonic acid, okay? Because you get rid of the coenzyme A. The thioester is cleaved by water over the bond, hydrolysis. And you get a ketotic dicarboxylic aciduria, and they could be detected in urine as well. There you go. Now I've explained the biochemistry of the system up to the physiology. So, what about some metabolic relationships with tissues that are associated with nutrition, hormones, and disease states, okay, like obesity? We looked at this before, but I just want to remind you that the obesity system, okay, that we're worried about here is going to be impacted by the malonyl-CoA decarboxylase, acetyl-CoA carboxylase rheostat. That's the key feature. So during an obese state, pancreas is producing insulin. Okay, you have a lot of circulating, you're hyperglycemic. So you got a lot of glucose, a lot of amino acids in the blood. The amino acids and the glucose are all going to get converted to lipid. So, and in fact, even lactate from the periphery is going to be converted to lipid. In fact, the lactate dehydrogenase, which we just talked about, that's where glucose is going to send its carbon. The lactate ultimately will also be used to synthesize pyruvate, and the pyruvate back in the TCA cycle to citric acid to make more fat. So VLDL can also bring lipid into the system. So you're starting to make a liver, which is becoming a fatty liver. It's not alcoholic fatty liver disease, for example. You're also getting chylomicrons that are trying to flood the viscera with more and more triacylglycerol. And so is the VLDL. So you're getting a lot of lipogenesis. You're getting a lot of lipid transport from the liver. You're getting a lot of storage of lipid in the liver. And you're getting an increase in the adipose tissue, visceral fat, depot fat. This is the obesogenic stage. Okay. So during diabetes, it's different. Diabetes, you get type 2 anyways. Insulin is generated. You're running, of course, amino acids and glucose into the liver. You're making some lipid. You're making some glycogen. But also, glucose is going to start accumulating because insulin resistance occurs because it's type 2 diabetes. So you're not going to get glucose uptake into the lipid, into the adipose tissue, excuse me. It's going to be blocked. So it accumulates and it goes back to the muscle tissue. So now you're starting to get heavy doses of glucose in the muscle tissue, which you know is insulin-dependent uptake, that burns out the insulin receptor. So you get insulin resistance because of it, because of the high levels of circulating glucose, because it can't make it to the adipose. Right? And it ultimately gets blocked in the adipose. Triacylglycerol also accumulates because you're making a lot of that through the VLDL pathway coming from the liver. Okay? And chylomicrons are also feeding into that. So you have now dyslipidemia, hyperglycemia, and you're starting to put lipid deposition all throughout the body, periphery. And that includes heart muscle and even in the central nervous system. So that's what diabetes is doing because it's association to obesity. Now, here's going back to this oncogene paper. Muscle cells use malonyl-CoA to control fatty acid oxidation. As we said, mal-CoA levels are modulated between the flux of those two enzymes. I've already mentioned the malonyl-CoA decarboxylase. Now we're going to look more at acetyl-carboxylase. Pharmacological MCD inhibition increases malonyl-CoA, which blocks beta oxidation because mal-CoA blocks that CPT1 pathway, remember? And that's what I say there, okay, CPT1. Now, ACC, the acetyl-carboxylase, this is this enzyme here, which normally converts acetyl-CoA to malonyl-CoA. Right? It requires ATP, and it's a biotin-containing enzyme which allows for carboxybiotin intermediate, so the uptake of CO2 via bicarbonate, which is hydrated, directly into the cell. Right? And you know that MCD carries out that backward reaction, just releasing CO2. Okay? So understand that this couple is really important, because once you make malonyl-CoA, in the presence of NADPH, fatty acid synthase will make palmitate. You steripide to CoA, you make palmitoyl-CoA. Long-chain fatty acyl-CoAs can go into the mitochondria for beta oxidation, but because of high levels of mal-CoA buildup, because of inhibition of the decarboxylase, what happens? You block long-chain acyl-CoA uptake, so you don't get fatty acid oxidation, but you can get ketone body oxidation, because that doesn't require CPT1. Okay, that's what's going on here. Right. So keep that in mind. So this comes from that particular website there, talking to you about acetyl-carboxylase regulation. You need to learn a little bit about that now. There's a reaction, ATP bicarbonate, that's hydrated CO2. ATP drives the reaction of the carboxylase. The enzyme itself exists as monomer. Now look here. You have an inactive monomeric acetyl-carboxylase, which is phosphorylated. Okay? So when it's in its phosphorylated state, it's inactive. Insulin will turn it into, okay, a monomeric acetyl-carboxylase of low activity. All right? Because insulin, once it's high enough, because you want to make lipid with high circulating insulin, it's going to give you a monomer that can carry out some carboxylation, make mal-CoA basically. Right? Now, if you want to make a lot of mal-CoA for a lot of de novo fatty acid synthesis, right, you need to be able to get palmitoyl-CoA, which promotes depolymerization, or you want citrate to promote polymerization. So palmitoyl-CoA will depolymerize acetyl-carboxylase. And when you do that, that's an allosteric control. You inhibit the massive production of malonyl-CoA because the enzyme isn't fully active. Okay? Likewise, citrate will promote the polymerization of the monomeric acetyl-carboxylase after it's been stripped of phosphate because of insulin, because of a phosphatase that insulin turns on, basically. Then you're going to make a lot more malonyl-CoA. So citrate leaving the mitochondria is a signal. There's a lot of acetyl-CoA coming in. Acetyl-CoA is going to be used for what? Yeah, de novo lipogenesis. That's just all is what they're saying here. Polymer is active, the monomer is inactive. Citrate induces a polymerization. Okay? And then therefore you get active lipogenesis because that's basically the rate-limiting enzyme, the carboxylase is. Insulin carries out the dephosphorylation. It gives you a that's how insulin gives you a monomeric active form because you got rid of the phosphate. That's completely dead on over here. But you're not on the way to making full-blown active carboxylase until you polymerize. Citrate's necessary for that. It's a multiple induction, right? Don't worry about this high-fat diet and fasting. We're going to get into that later. But that's basically a repression phenomenon. So you decrease the amount of de novo fatty acids on a high-fat diet. So inhibition of de novo fatty acid synthesis can induce apoptosis of cancer cells. So if you block fatty acid synthesis in cancer cells, you can cause those cancer cells to go through programmed cell death. Since MCD co-regulates the level of mal-CoA, which of course performs multiple tasks in the cell, including bioenergetic balance between fatty acid synthesis and oxidation, what I just showed you for the last hour, treatment of human breast cancer MCF7 cells with an MCD small interference RNA or siRNA tanks ATP levels and proves, cytotoxic to those cancer cells in vitro, presumed by blocking beta oxidation. So those cancer cells that grow actively and eventually metastasize would much rather use fatty acids as fuel as glucose. That's the point. However, follow through here. Synthesis and pharmacological characterization of a small molecule inhibitor of MCD, so they made a small molecule inhibitor, which is this 5-morpholino, 4-carbonyl, 4-222-trifluoro, 1-hydroxy, 1-trifluoromethyl-ethylphenylamino pentanoic acid methyl ester, or you can call it MPA, reveals inhibition of MCD activity in those cancer cells, increases cellular mal-CoA, and yields cytotoxicity to a number of human breast cancer cell lines. That's good, right? Now, however, given what we know about the Warburg effect of rampant aerobic glycolysis, or glycolysis in issuance of high levels of oxygen, in a tumor cell core as opposed to the periphery of the tumor, this effect may hinder MCD inhibition in certain stages of metastasis and growth of primary tumors, since mal-CoA built up with block beta oxidation, thus forcing a switch to glucose as a bioenergetic substrate, thus generating the Warburg effect, thus fueling the core, the medulla of that whole tumor complex. So you see, you have to be very careful when you're considering what to block and what not to block. Right? One more little thing. Remember that when ATP depletes, AMP kinase turns on. Remember what it does to AMP kinase turns on enzyme phosphorylation, which will enhance beta oxidation, but it'll block all those de novo pathways. Gluconeogenesis, protein synthesis, that's usually through the mTOR pathway, AKT-mTOR pathway, if you want to know. Tanks lipogenesis, both transcriptionally and also in control of enzyme activity. And now, of course, the other lipid in membranous cholesterol is blocked. So here's a paper that I'm going to finish with, Cell Metabolism, published again just 6 months ago. Sorry, get my ugly mug out of the way there and show you. Okay, here's your full citation. HCC is a disease which I've talked a lot about in Vera Mad and in Authentic Biochemistry lectures, as I'm sure you're aware. HCC is a disease that's hepatocellular carcinoma with a high mortality. It's greatly increased with obesity and with NAFLD, or non-alcoholic fatty liver disease. Of course, alcoholic fatty liver disease will also enhance HCC. So NASH, which is non-alcoholic steatohepatitis, which is an inflammation of the liver because of high levels of lipid deposition. That's what NASH is. And the NAFLD, non-alcoholic fatty liver disease, which we've talked about extensively in this system. They're linked to hepatic dyslipidemia, of course, and that can lead to cirrhosis, ultimately to the carcinoma. All right, bad news, right? Yeah. Functional loss due to known point mutations within the AMP kinase phosphorylation sites of the ACC. So it's not the AMP kinase that's got the mutation, it's the ACC1. So serine 79 goes to an alanine, or ACC2, serine 212 going to an alanine, can't phosphorylate an alanine, right? Have been linked to aggressive, if you can't phosphorylate it, you can't control the carboxylase. You see? So you get aggressive de novo lipogenesis because the carboxylase is always on. It can polymerize, right? There's enough citrate. And that's what happens in cancer cells. So a novel liver-specific inhibitor, that's something I call ND654, that mimics the effects of the ACC phosphorylation, interestingly, inhibits hepatic de novo lipid synthesis and the development of the carcinoma, improving survival of tumor-bearing rats when used alone in combination with a multi-kinase inhibitor known as sorafenib, because that blocks all those kinases. Typical drug use. It's an inhibitor drug. These data and accompanying corroborating research that these authors in this paper did in their colleagues in other labs, that research evidence suggests that de novo lipogenesis and dysregulation of the AMP kinase-mediated ACC phosphorylation and accelerating the cancer could be a pharmacotherapeutic agent. All right, that's basically what it's showing you. Here's this drug. All right, it's going to block this whole phosphorylation cascade here. And because of that, you're not going to get the acetyl carboxylase. You're not going to get de novo lipogenesis. So you don't want to make it go to a hepatocellular carcinoma, right? You want to block that pathway. And that's what this is trying to do, block it. Okay? So it's mimicking that normal phosphorylation, right? Mimicking it. Remember when you get a phosphorylated ACC carboxylase, acetyl carboxylase, excuse me, you decrease its activity. Right? That's what happens. Right. So you get hepatocellular carcinoma with this system, normal liver, you do not have de novo lipogenesis. That's what this is all showing you. All right. So we're going to stop there. I'm not going to finish with these other hormonal regulatory pathways. That's for another talk. Now, what have we done today? I showed you the malonyl-CoA decarboxylase and acetyl carboxylase, what I call a rheostat, an on-off switch. Okay? That tightly regulates the bioenergetics of a cell, either a cardiac muscle or a tumor cell, the two I looked at. And it does so differentially. And even within one system, it does so differentially because of the core or the early stages of a tumor growth, which can become anaerobic and rely on rampant glycolysis, whether or not there's oxygen, right? Or fatty acid oxidation, which can then generate a metastatic tumor system later on during the age of the tumor. Okay? And so inhibiting MCD may be a good thing to do later, because early on you're having this rampant glycolysis, depending on the kind of tumor it is. But with the heart disease, remember after ischemia, you want glycolysis. You don't want lipid oxidation. And one of the reasons for that is a lot of molecular oxygen around will generate a lot of partially reduced forms of molecular oxygen known as ROS, reactive oxygen, further enhancing the probability of another heart attack. So the take-home message is you cannot derive a pharmacotherapeutic signal into one or two gene products like an ACC or an MCD and expect to have an entirely healthy system in a human. You have to consider a lot of different disadvantages of blocking those pathways, including what happens at the transcriptional level. That's why pharmacotherapy is a very complex field, and that's why pharmaceutical scientists are always working for drugs which are more specific and which can be turned off and on very easily, rather than having suicide inhibitors, for example, having competitive inhibitors for enzymes. Or if you're doing something in transcriptional control, having something that loses its control immediately after chromatin remodeling, or the inhibition of chromatin remodeling, or thereafter. It's like Wale. All right. I think I've said enough today. I think you get the idea of why I did this talk. Basically, I wanted to get you inured into this whole idea that in biochemistry, that if you have a biochemical system, and all you need to do is turn something on or off, that you're going to get back to having a healthy recovery. That's not always the case. It's very complex because the system is perfectly balanced for the cell, and for the tissue, and for the organ system, and for the whole body. That's called homeostasis. When you start turning something off or on with subtle changes, sometimes you can tip the entire system over and go from a cardiac muscle disease to maybe even generating something like a cancer, or from controlling cancer and then inducing a cardiac muscle problem that can be induced by the drugs you're using. Right? All right. So with no further discussion than that, I'm going to end. Hope that you enjoyed this lecture today on this fine 4th of June, 2020. This is Dr. Dan Guerra saying bye for now.